Biopsychology is a broad and diverse discipline that studies many different phenomena and approach their research in many different ways. In this module, we will look at the different types of research that characterize the biopsychological approach. Because of the complexity of behavior and mental processes, biopsychological research varies in three dimensions: subjects, methods, and types of research. Throughout the next slides, we will be looking at the pros and cons of doing research with human and non-human participants. We will look at the difference between experiment and non-experimental designs, and we will look at the types of research, differentiating between pure and applied research. Before going in detail on the pros and cons of doing research with human and non-human subjects, it is important that we understand evolutionary continuity of the brain, which is the reason why we study non-humans. Most commonly, we study mice and rats. However, there are other species that have also been studied. And the reason why is because much has been learned, and we believe there continues to be a lot of potential in studying the brains of other species, because species differences are often more quantitative than qualitative. That is, that the cells, specifically the neurons and the glial cells in the nervous system, are similar, very, very similar. And in fact, the only difference that exists between the cells in a rat versus the cells in a human is that humans have more. And that's why there's more higher quantity there, but the quality would not be compromised at all. Looking at the reasons why human subjects are often preferred by some, depending on what we're studying, is that human subjects are capable of following instructions clearly. For the most part, I should say. But a rat or a chimp or any other species may struggle to comprehend very complex instructions, whereas a human typically is capable of following that, whether they be just stated or in written form. Additionally, human subjects can also report their subjective experiences. So as we expose them to a particular stimulus, the human may be able to then report back on their feelings, their emotions, and any sensations they may have experienced during the study. Believe it or not, humans are often cheaper to work with than animals. And the reason why is because of the cost of maintaining an animal lab, which can be very expensive, as there is a need to have employees there for extended periods of time. We have to feed them on a regular basis, and this can become very expensive. Whereas working with humans typically, or may sometimes work based on the appearance of the human once, and then thereafter we dismiss them, then it is up to the researcher to then analyze the data, but they're not required to feed them or to take care of them. And perhaps the most important benefit of doing research with human subjects is that humans have a human brain. And although we stated earlier that evolutionary continuity allows us to see that there's more of a difference of quantity than quality, we do see that not all studies that have been discovered in rats hold true with humans. Although there may be some similarities, the propensity in humans may not be the same as in rats. For example, there was a study in which rats who were fed cereal on a daily basis developed diabetes at a rate of 7 times more than those rats who were not fed cereal on a regular basis. When this was tested with humans, the rate at which individuals would develop diabetes was not the same as with rats. It wasn't necessarily denied, but it wasn't at that particular rate. And so we understand that although there may be similarities between non-human and human subjects, only with human subjects can we then get a better picture so that we can generalize our findings. Working with non-human subjects has many benefits, and although it has been controversial because of the belief that some animals are harmed and hurt throughout the processes, the APA, the American Psychiatric Association, and the American Psychological Association mandate that animals be protected from mental and physical harm as well. Having said this in here, animals, non-human subjects that is, typically have simpler behaviors and brains, which make it more likely that a brain-behavior interaction will be revealed. For example, when we look at an animal that seeks to engage in sexual behavior, there aren't any social or religious or moral issues at hand. Typically, if they feel aroused, then they simply will do it. Whereas when we look at humans, because of societal demands, because of moral and religious reasons, we are not able to demonstrate the behavior as clearly as an animal would. This complicates the understanding of human subjects and facilitates the understanding of animals. We also find that our insights with non-human subjects typically arise from what we call the comparative approach. And this, again, is making comparisons with other species. For example, the cerebral cortex. As we look at the cerebral cortex of different species, we find that there are different parts of that brain, that part of the brain, that control parts of their body. As we look at an elephant's brain, we find that a significant portion is devoted to the trunk. And the reason why is because elephants use the trunk to eat and to protect themselves and to even clean themselves. And so as we go through the process of comparing that to a human brain, we would find that there would be no significant portion designated for our nose, which may be the equivalent of that. However, because our hands are responsible for doing the task that a trunk of an elephant would, we find that that same portion of the human brain would be devoted to hands and feet. With non-human subjects, we also find that it is easier to work with, although, as stated earlier, we aim to protect the animals from mental and physical harm. We do have more freedom to conduct certain studies as long as we are able to demonstrate that the benefits of the discoveries of such studies would outweigh the consequences. We may be able to get approval to conduct certain studies. We will now look at the second dimension of doing research in biopsychology, and that is the method design. During experimental design, we are able to determine cause and effect relationships. This is done so by making use of a representative sample that would then be used to make an inference about the population at large. You may already know that it is nearly impossible to get every single human to respond to a survey or be part of a study, as it would take a lot of money, a lot of resources, and a long time. However, by getting a large enough sample size, we could then discover differences and behavior and mental processes that are consistent across society or the human race. When asking a question in here, and doing an experimental design, we have two types of experiments. We have either between subjects design or within subjects design. The between subjects design is a design in which a different group of subjects is tested under each treatment condition of an experiment and tested only once. For example, if I wanted to see if the effect of caffeine helps students score better on an exam, I would have to divide class into two groups, and one group would be given caffeinated substance, whereas the other group would not, and then I would compare the results. However, we also have within subjects design, and this is where the same groups of subjects is tested under each condition, meaning that I would not separate the class into two groups. Instead, I would test you all in one day under the influence of caffeine, and then on a separate day, the exact same people without exposure to caffeine. These have their pros and cons, and we'll discuss a little bit more about these in the future. But for now, I would like to establish also the variables that are required in experimental design in order for us to understand whether in fact caffeine has an effect on test scores, we have to identify our variables. And we have in experimental design the independent variable and the dependent variable. Please note that the independent variable is that which a researcher is able to manipulate, and this is a hallmark of the experimental design, that we are able to manipulate and administer a variable, in this case caffeine, at will. The dependent variable is that which depends on the independent variable. In this case, it would be test scores. If in fact consuming caffeine has an effect on test scores, then we would be able to see that through research. Putting this into practice and to understand the population at large, we would like to assess whether the type of dog has an effect on the level of fear. We could expose you to this dog here and see if in fact there is a level of arousal that would be high. And after assessing your stress level and cortisol levels, we can also assess the same if you were to be exposed to this dog here. If in fact there was a difference in your arousal level, depending on whether you were exposed to this bigger dog or the smaller dog, we would be able to determine cause and effect relationships, as we would have manipulated the type of dog, which is our independent variable, and then we would measure your arousal level or level of fear, which would be our dependent variable. Please note that this is the only method that allows us to demonstrate cause and effect relationships. Although conducting an experiment seems conceptually simple, it is often difficult to practice because we have to ensure that there is only one difference among conditions. And when working with subjects, oftentimes this is very difficult, as there can often be what is known as confounded variables or confound. These are unintended differences between conditions that can influence the dependent variable. For example, looking back at the type of dog and the level of fear that you would have experienced had you been exposed to those dogs, those two different types of dogs, we would have to take into account whether you are a dog lover or whether you own a dog. We would also have to take into account whether you've ever been attacked by a dog. And those things, as much as we may try to control everything in the experiment, we may not be able to. And so, because these are difficult to eliminate, these confounding variables are difficult to eliminate, they can make the experiments difficult to interpret, as it may be difficult to tell how much of the effect on the dependent variable was caused by the independent variable and how much was caused by the confounded variable. An interesting example of how confounding variables can be difficult to spot come from our 21st president, Calvin Coolidge. During his presidency, he was touring poultry farms as an attempt to stimulate the economy. And while being there with the First Lady, Mrs. Coolidge inquired of the farmer how his farm managed to produce so many eggs with such a small number of roosters. The farmer proudly explained that his roosters performed their duty dozens of times each day. Miss Coolidge then asked, "Perhaps you could point that out to Mr. Coolidge." The president promptly asked the farmer, "Does each rooster serve as the same hen each time?" The farmer replied, "No, there are many hens for each rooster." The president said, "Perhaps you could point that out to Mrs. Coolidge." These apparent jabs seem to have gone beyond this exchange, if in fact it took place, though I've read about this and it's in your textbook. I have not come across any sources that are able to confirm that this exchange actually took place. However, the question here was whether a copulating male, in this case a rooster, who becomes incapable of continuing to copulate with one sex partner can often recommend copulating with a new sex partner. And though they found that to be the case with males, they wondered if females were able to display the Coolidge effect. Because the males of most mammalian species become sexually fatigued more readily than do females, the attempts to demonstrate the Coolidge effect in females are often confounded by the fatigue of the males. Lester and Grzelka argued that the Coolidge effect had not been demonstrated in females, not because females were not capable of, but because of the effect of the males. And by effect, I mean the fatigue of the males after copulating once. While some researchers believe that females were not capable of demonstrating the Coolidge effect, Lester and Grzelka were not convinced. So in 1988, they were able to study whether in fact female hamsters were capable of demonstrating the Coolidge effect. Lester and Grzelka did this by measuring the amount of minutes that female hamsters engaged in lordosis pose. Lordosis is when a female hamster has an arched back and a rump-up-tailed, it's diverted posture, which is an indication of sexual receptivity. And what Lester and Grzelka were able to demonstrate was that female hamsters were more sexually receptive to an unfamiliar male than to the male they had already copulated with during their earlier test. They did this by separating two groups, and so we have the unfamiliar group and we have the familiar group. Now, what Lester and Lazarga were able to do was to expose females to a male, and you can see that whenever both females from the unfamiliar and familiar group were exposed to the first male, they engaged in lordosis time of 30 minutes on average with the first male. However, the second time they were asked to copulate in the unfamiliar group, there was a reduction, but was similar nonetheless. With the familiar group, the female hamsters were exposed to the male they had already been exposed to previously, and you can see in here that the amount of time they engaged in lordosis was significantly less than the unfamiliar group who was exposed to a different male. This allowed us to understand and confirm that in fact females too are able to demonstrate the Coolidge effect, and it is not just males. However, for many, many years, there was a belief, a conception, that it was only males who would have these particular weaknesses, and which would allow them to then copulate with multiple females. And by understanding the research in species such as hamsters, we've been able to understand behaviors that continue to puzzle us. For example, as though there is a debate as to whether pornography addiction is real or not, we do find that individuals who engage in watching pornographic material for extended periods of time, often interfering with their daily functioning, have high levels of dopamine released when they are exposed to different women. And this is what allows people to go on time and time again watching the same content, the same sexual content, as long as it is a different phase or a different setting. Oftentimes, these men would neglect sleeping time, and oftentimes are as exhausted as the poor hamsters. Although the experimental design is the only one that allows us to demonstrate cause and effect relationships, sometimes it is impossible to conduct controlled experiments because of ethical and technical reasons. So when we are unable to, we then engage in non-experimental designs. The two types that I would like us to focus on here are the quasi-experimental studies and case studies. Starting with quasi-experimental designs, please note that participants who are involved in studies like these, they decide which group they would be in because of a predetermined criteria. For example, if I wanted to research whether left-handed people are significantly more artistic than right-handed people, I cannot randomly assign people to either the left-handed group or the right-handed group. I would have to ask individuals who are left-handed to then, by their natural setting, be in that group, and those who are right-handed, because of their natural setting, go in the right-handed group. This would not be possible in an experimental design, as the experimental design mandates that we assign people to the two different groups in a random fashion. If a researcher wanted to study whether alcohol intake has an effect on brain, and more specifically brain damage, we would not be able to do an experimental design, as it would require that we administer alcohol to the individual just to determine whether in fact it causes brain damage. However, that does not mean that we cannot study alcohol intake's relation to brain damage. Quasi-experimental studies allow us to study this in here by conducting studies with individuals that would have already exposed themselves to alcohol intake, and if they identify as alcoholics, they would then be participants in our study, and we'd be able to perhaps assess recall capacities and IQ tests and various other skills that we'd be able to study in comparison to those who would not be alcoholic. Although it may seem like an experiment because we have two different groups, please note that there are many confounding variables that are not controlled. As with alcoholism, you may already know that typically that is a symptom of another issue. And perhaps the reason why they're alcoholics is because of childhood trauma, because of romantic issues in the past, or what have you. And although we would claim that all participants were equal, we would perhaps fall short of taking into account the various differences that exist between the two. The other form of non-experimental design is a case study. This is a focus on a single case or a small group of people that allows us to understand the individual in question. Freud himself used case studies to develop his theory, and this is one of the reasons why his theory, the psychodynamic theory, is often criticized because of lack of validity for people other than those he worked with. Case studies allow us to do an in-depth analysis of the individual, and while it is a good source of testable hypotheses, one of the major problems is generalizability. And by generalizability we mean the ability to apply our understanding of that individual to the population at large. For example, Jimmy G, the case that you may have read in your textbook, is a man who was unable to form any lasting memories since his early 20s. Now, by understanding his childhood and understanding his activities and whether he was exposed to environmental places where there may have been toxins that affected the brain, and as much as we discover about him, the reasons that kept him from forming lasting memories may not be the same for everybody else. For example, if you think of people who perhaps consume caffeine, although all humans may be able to consume caffeine, not everyone responds the exact same way. Therefore, our understanding of one individual in depth does not ensure that we understand the population at large. Last, we'll look at the third dimension of biopsychological research, which is the type of research. Now, we'll be looking at pure and applied, but before we do so, I'd like to ask you whether if you were part of a committee that was in charge of voting for the use of federal funds, would you vote in favor or against the use of $150,000 of federal funds to investigate what maggots prefer to eat? Now, if you think about this in here, these are the maggots, and these are typically not very nice species, very nice creatures. People may be more inclined to vote yes. Had this been a dog, people would have perhaps been more inclined to vote yes had it been a dolphin. But when we look at species that are so expendable and not necessarily pet-like, people often vote on emotion. And so I'd like you to consider this in here, because in order to understand pure and basic research, you have to understand that pure research is often more difficult to start and to even get funding for than applied, because the benefits are not necessarily obvious. Investigating the nutrition of maggots has given way to maggot therapy which is a type of biotherapy involving the introduction of live disinfected maggots into non-healing skin. For example, individuals who have wounds because of a amputation or a deep laceration often get infected, and because of the infection or the severity of that, oftentimes the antibiotics do not do a good job at cleaning this in here. And by researching what maggots like to eat, they discovered that they actually enjoy eating and they feed off of dead skin and dead tissue. This has benefited a lot of our servicemen, a lot of our veterans who unfortunately have come back sometimes with amputated arms, and because of the procedure, sometimes they have to continue amputating higher and higher because of gangrene. And now what we're able to do is get maggots to be placed in a plastic gasket by their arm or their leg, wherever the wound is, and then the maggot would be in charge of eating the bad flesh. This, as you can see, is one of the benefits of doing pure research. However, it was not very clear when it started. Pure and applied research are typically defined by the motivation of the researcher. As stated earlier, individuals who would have been driven by wanting to acquire knowledge about maggots would have possibly not known that maggot nutrition would have benefited mankind, specifically with lacerations and infections in the flesh. However, once we have some understanding of some species and some basic elements, we could then begin applying that so that we could bring about some direct benefit to humankind. Please note that pure research is often more vulnerable to political regulation because politicians and the voting public have difficulty understanding why research of no immediate practical benefit should be supported, especially when we go back to the question earlier of deciding whether $150,000 of federal funds should be allocated for the investigation of what maggots like to eat. However, please note that oftentimes research is done so by individuals with the intention or purpose of acquiring knowledge. Doing so allows individuals to then find better ways of treating things, alternative ways, and we hope that scientists as a whole would not be driven by greed and fame, but by a genuine desire to help improve society or mankind. Jonas Salk, who was the guy that developed the polio vaccine, was asked if he would be interested in patenting the polio vaccine. And his response to this was, "Could you patent the sun?" We hope that there are more scientists like Jonas Salk who, rather than seeking fame, were simply just interested in acquiring more knowledge for the benefit of everyone.